So welcome everyone for our virtual seminar on neutrophils in chronic inflammatory and autoimmune diseases. Maybe a little bit background to our seminar series. This is a product of the pandemic, because we realized usually we have larger visiting professorships where people come by, and this was obviously not possible during the pandemic. And so we thought for a replacement and had organized in the meantime, I think 2 or 3 of that online seminars to replace that visiting state-of-the-art in translational rheumatology seminar series. And it was a great success because we realized that even more people can join around the globe. So this is now becoming an international seminar series that is overall well attended. We are very grateful for the speakers that we are inviting. And this is sponsored by pharma companies. They are paying into a pool of support, so they don't have any influence what we are supporting. You're seeing this currently in the sponsors, and we're very grateful for them that they are supporting education, scientific education, and let us in particular choose what we want to talk about. And along this line, we have decided to do a seminar on neutrophils today. And that was greatly organized by Caroline Ostfeld, Kinga, and Nicole Schneider. And we are very grateful and thankful for them to help us to set this up. Caroline Ostfeld will take over the second part now from Mariana Kaplan and lead the discussion and introduction of Mariana. And it's now my pleasure finally to introduce Martin Herrmann. Martin, probably remember that I studied in Erlangen, actually my medical studies. So I know you for, I think, 30, at least maybe 35 years.
I am aware of this, yes.
And Martin is for a long time a very successful senior PI in the Department of Rheumatology in Erlangen, has made important contributions to the field of experimental rheumatology. And Martin, we are very grateful for you to now be so flexible and join us on the first talk today. And the floor is yours, Martin.
Hello, everybody. It's a real pleasure to see very old friends in the auditorium, and which have not seen for the last 3 years live. And one of it is Pierluigi and also Brigitte Gulstadt. And very pleasure to you. I was contributing to rheumatology when Calden was the head of our department, and then I changed a little bit my topic, and I went to the neutrophil diseases in the Acute of shed. And I will now present some data on the bad news from neutrophils, especially neutrophil extracellular traps. But I have to stress in the beginning, neutrophils are good cells. Nets are good functions. Neutrophils are required for life. This is much more pronounced in humans than in mice. Mice can survive a certain time without neutrophils. Humans cannot do this. Humans need neutrophils and also net formation. But unfortunately, everything which is so needed for everyone, there may be some mistakes going on and some misleadings of the activity. And this I will stress today. But neutrophils are good guys. I have to switch the. I don't know how to switch. I cannot switch my talk. I cannot. Oh, yes, it does. So the disclosures are some disclosures here which are related to the field of the neutrophils, and as you see here, there's still space for some Swiss companies here to join my portfolio of disclosures. Now I go to neutrophil function. Neutrophils are cells which have a very nice nucleus, which is very decisive. If you look for the microscope, you see the nucleus, you can easily detect neutrophils. But that's only true for the neutrophils when the neutrophils are in the normal or in the activated status. It's not anymore possible for neutrophils which have undergone net formation or apoptosis. You see the nucleus. Why is the nucleus shaped like it is? The nucleus is lobulated because then the cell can go much faster through tissues because the nucleus is not the diameter of the nucleus is less. So the neutrophils are very fast cells, and the multilobular nucleus allows fast speeding up of the neutrophils in the tissue. The neutrophils carry antimicrobial agents. They are ready to phagocytose bacteria and fungi. They also can produce the ROS, an atomic bomb basically for the tissue, which destroys all pathogens, but unfortunately also the tissue of the host. And the last developed activity is the formation of nets. And neutrophils only have two different appearances. One is a normal regular neutrophil, and the other one is a neutrophil in the beast mode. The beast mode is basically described by Mariana, Kaplan. It was neutrophils with low density. They show up in the, if you isolate cells, they show up in the PBMC fraction and not anymore in the PMN fraction. And Mariana, I guess, will talk about this later. And there are good neutrophils, which are anti-inflammatory. As you see here, you can see it on the nucleus, and there are pro-inflammatory neutrophils here. And they are circulating, and both of them have their function. This is the anti-inflammatory neutrophil is basically living in a steady state condition, and the pro-inflammatory in disease. Now, what is going on when you put small nanodiamonds to neutrophil suspension? You see the blue nucleus is a closed nucleus, and after 7 minutes, the video is 7 minutes, the neutrophils throw out their DNA, and now it's getting red. That means the DNA is now accessible to propylium iodide. And this is the net formation, the fastest net formation I've ever seen. And this is done by the neutrophils' best stimulants, diamonds. And the diamond works as an soap. It just soaps the neutrophils, and then they pop open and throw out the DNA into this vicinity. And now you see it here. This is the seconds of the movie. Now, something important. It's not so easy to stain nets. It's easy to stain nets in vitro cultures. It's very difficult to stain nets in certain viable tissue sections. And so you see here, this is a cells where you only see the nucleus, and you see here when the nucleus of the cell is a good red stain, the net, which is shown here, yeah, with the arrows, the net is very faint in staining because the DNA, which is condensed here in the nucleus, is spread all over the tissue, all over the culture in this condition. That's not so important for in vitro cell culture. You can find in vitro cell culture, you can find a condition to stain it properly, but it is difficult for the tissues. So DAPI signal for spread extranuclear DNA is rather low, and this precludes high sensitivity detection of nets in tissues. What to do? What we observe here is a tissue from a patient with a tumor, and the patient has an extended field of net formation in the tissue. You see here, you see the overexposed nucleus here, which is seen in red, which shows that there is DNA, and we have to overexpose, otherwise we would not see this background staining, which usually is considered unspecific. However, when you stain that tissue with an anti-DNA antibody, which is usually found in lupus patients, or you can also make it from gene technology, you see that the DNA in between the nuclei there is really DNA. It's not just a background signal, which is shown here. And to confirm that it is really chromatin, we also stained the same section with histone. And if you see, the histone is giving the same picture as the DNA. And as you see here, you see also widespread neutrophil elastase, which clearly shows that this area of the body contains net formation. That means if you stain the DNA, anti-DNA antibody. This is, by the way, IgM. You can clearly detect, preferentially detect the DNA outside the nuclear region. DARPI and PI stain any double-stranded DNA, the nucleus as well as the extranuclear DNA, but anti-DNA IgM prefers the decondensed chromatin of NETs, and that's why you can make it much better visible in tissue sections. Now let's go to the acute pancreatitis model we investigated. What we did is we injected IL-17 Viline-Cray. I have to remove this here. Yeah, to Viline-Cray, and you clearly see that there is we intended to produce IL-17 in the intestinum and do some colitis or so. But what happened? We got pancreatitis. We did several different ways to induce IL-17 with hepatic and other vectors as well. We never got another thing than pancreatitis. And so pancreatitis is characterized by the presence of these structures, which is original figure from our paper here in the pancreatic ducts. And you see DARPI and as well as MPO is clotted together here in the pancreatic duct. And you also see that this is citronylated, and you see here's a citronylation of the same consecutive slide. That means in the pancreatic duct, you clearly see some clotting, and this, of course, is not a good idea for the pancreatic physiology. Pancreatic ducts of IL-17 transgenic mice are concluded with aggregated nets, and PAD4 knockout mice are completely protected. If you take out the PAD4 gene, the mice can still undergo net formation, but they cannot spread the net as good as they spread it in the wild-type mouse. This is my video for this. This is a duodenum, which has few bacteria in the duodenum. There's a pancreatic duct. This is the acinus, and here's the neutrophils. And as we observed, the bacteria may enter that duct, yeah, but the neutrophils come in, form nets, and like a cleaner for a saxophone, they wipe out the bacteria back into the duodenum, together with the fluid of the pancreatic duct. So what happens if you have too many neutrophils in that condition because of IL-17? You get net formation, and now the nets are not anymore able to wipe out the bacteria, but they clot together and occlude the pancreatic duct. What is the result is there is a pressure distal, proximal to the acinus, and there is also the bacteria cannot be wiped out. So there is also inflammation here due to bacteria, and here is due to pancreatic digestion, self-digestion with the pancreatic fluid. And all this together leads to a pancreatic phenotype, which we observed very stable in mice. If you have now PAD4 here, the nets here are smaller, much smaller. They are not able to felt to each other, and they do not occlude the duct so properly as in wild-type conditions. Nets protect pancreatic ducts from bacterial invasion, and aggregated nets may occlude pancreatic ducts. Let's go to cholelithiasis. Here you see this is an epifluorescence photography of an gallstone. Here, all these different forms of gallstone: the white one, the black one, and the brownish one. And if you look here to the epifluorescence microscopy, you see that on the surface of the stone, you see patches of DNA which are on the surface of the stone on all stones we have investigated. And we next investigated several gallstones, many of these from a museum in Berlin, which are already 10 years out of the body, for neutrophil elastase activity, which turned out to be extremely stable enzyme in this condition. This is not much in the center. The center is the oldest part of this gallstone, but there is still activity. It's in this paragraph is not seen very well, But in the body of the gallstone and on the surface, you find high amounts of neutrophil elastase activity, showing that there are DNA patches as well as nuclease neutrophil elastase activity, and we conclude that this may be related to the formation of nets. Gallstones carry DNA patches on their surfaces, and gallstones harbor neutrophil elastase activity. Even, of course, we also investigated new stones, yeah. But in this case, shown in the picture, we only have stones from the museum. Now let's turn a little bit more up. Sialolithiasis. This is a material taken out of the duct of the salivary gland, and you see here there's almost none of the red stained nuclei, which is DNA stained by PI, is anymore in a normal shape. Yeah, there's almost everything is already wiped out and getting at least necrotic. And here in this part, there is the stone. And the stone, inside of the stone, you also see this DNA, which is accumulating inside the stone. And when you now look for other markers, you recognize that all these salivary stones are made from gnats, and some of them even from gnats made from eosinophils. And this is a neutrophil elastase of the same picture. And as you see here, if you're aware that neutrophil elastase is usually inside a cell, or it is in granular in the tissue. And after degranulation, for example, this small piece here could be one of these, yeah. But here you see that the neutrophil elastase is not forming normal structures, but this is forming these net-like structures, which are made from the DNA, as you see in the picture before. And this is your overlay. You see here that the DNA is decorated together with the neutrophil elastase. You can also see it here. And here you see this rim around the stone, which you clearly see that there's a center with a neutrophil elastase, and there's a neutrophil DNA, and this was where the stone was. This is just a summary of the picture. And this shows you the structure here, which is very reminiscent of the net structures usually seen. Let's go even further up to the meibomian gland in the eye rim gland, which is providing the oil for the tear film. And if you do not have meibom in your tear film, it will evaporate and give you dry eye disease, which is a rather bad disease because it hurts in the eye, and this is incredible. You cannot see anymore. It's really a bad condition. Sounds nice, dry eyes, but it's not at all nice. In Germany, approximately 1 million people suffer from dry eyes. If you now look for the meibomian gland, you see the orifice here. This is a place here. It's a bigger magnification. You see the orifice of the meibomian gland where the oil should come out. The meibom, which is here in the backyard, and you can clearly see that this is occluded by structures which contain neutrophil elastase as well as DNA. So that we can say also the duct of the meibomian gland is occluded with the nets. And the problem is, yeah, it's not only that the oil is missing, but there's also pressure in the back. The acini produce meibom as they should do, and there's pressure in the system. And now the meibomian duct is widening very much, and this is really painful and needs sometimes surgery or even replacement. You see, although there's a neutrophil elastase, you see that there's citronylation in these glandular structures, and this is a negative control of the antibody. Now let's go to vascular occlusions. Chronic expression of cholinesterase factor 3 induces murine neutrophilia. That's known before. But if you have a human neutrophilia, it doesn't matter. The wild-type mouse and the DNase1 knockout mouse, they don't care about this. They are still healthy and everything is fine. But if you knock out DNase1 and DNase1 like 3, both of them, you see that the mice die within 4 days. And the question was, why do they die so quickly? And this one is a double knockout mouse. And if you add to the double knockout mouse a control vector, they die. In this day times it takes 5 days. And if you add a vector with the DNase 1 like 3 or DNase 1, either of these can prevent the death of the mice in this condition. And also when you induce a sepsis with LPS and E. coli, you see that there's a strong, there's a much of the mice dying in a couple of hours in this case, and only the DNase 1 and DNase 1 like 3 double mice, wild type mouse, they are protected from that death. That means in the condition of the sepsis in this condition is much worse in the presence of the double knockout of DNase 1 and DN1 like 3. And what we see, yeah, that here we take different things. We take crumb as a marker, and we take chromatin, and we merge it. We see occlusion of the pulmonary vessels in this mice, and you see also there's citronylation as well here inside the vessels. So that means we conclude that also in this case, these structures are occluded in the absence of the clearance mechanism of DNase 1 and DNase 1 like 3. The septicemia or the neutrophilia cannot be survived in mice. Neutrophilia cause high mortality in DNase 1 like 1 and DN1 like 3 double knockout mice, and this can be prevented by either DNase 1 or DNase 3 as 1 like 3 as an transgene. What is with the classical inhibition? The thrombin inhibition. If you add a thrombin inhibitor, dabigatran, it does not help any mouse to survive. It is independent of the dabigatran, and it's also independent of platelets. I did not show that picture, but there was always an enigma in the patients with sepsis. Where people told you, yes, there's a coagulation problem, but we always do anticoagulation, and it does not work, and we do not understand, and it doesn't work. And I guess in these conditions, the non-canonical attack against net formation is more promising than the classical canonical anticoagulation, which is anyway taken from any intensive care doctor to treat the patients. But they seem not to be very successful with the classical anticoagulative treatment. Neutrophilia-related coagulopathy is independent of platelet and thrombin. I have only one picture of sepsis included because it's now humans, and you see also in human sepsis, you see masses of neutrophil elastase in the vessels or in this pulmonary vessel aggregate together with a let's say spread DNA in the vessel. So that means also in the sepsis you see this in human sepsis you see this condition. But now I go to COVID-19, and as you all know, there's a coagulopathy in the patients. And what we collected, we collected lungs from the patients because of the clear problem with the and I thought that the lung is the airspace is occluded by nets. That was my hypothesis. And when we made sections here, you see this section, you see this structure could be either, but we stained it with CD31. It's a vessel. This vessel is completely occluded, and you see these vessels, all these vessels are occluded. Almost all vessels in this picture, not this one. Here, you also see DNA inside, but not neutrophil elastase. Many of the vessels you see here are occluded. And we also have patients where we did not find a single open vessel in the biopsy. By the way, this is autopsy. It's a patient who died from COVID. And you also see it here. It's full of cells in the vessels, and here as well. This is completely full, and this is very stuck together. And And all these contain DNA, and together with neutrophil elastase, which is at that time was our standard staining for the net formation in the tissues. Vessels of various sizes get clogged by net-driven non-canonical thrombi. And you see here, this is, I will now, this one is a DAPI, an A staining of the lungs of the patient, and this, it just take away the DNA stain, yeah? And this is a stain for neutrophil elastase alone. And you see everything here, yeah, is occluded. And when you look carefully at the picture with the CT31, you see that most of these structures are vessels. And these vessels are all occluded. And of course, this lung cannot breathe anymore. And it's not only that it cannot breathe, but you also see that close to the area of the big clot here, this is we call it maxi clot. You see that here the CD31 staining is abrogated because the endothelium is completely destroyed by the structure. Endothelium should look like this here, a ring which has no gaps. But this already means vasculitis as well as vascular denoculation, and that's basically deadly this condition for the vessel. Here, see it. And now we check the vessels. Here the normal healthies. Yeah, of course, normal healthies, you don't usually get lung biopsies, but this is the part of a biopsy from a tumor patient who has so the pathologist told it this is a healthy part of the lung, and you see the small vessels are all open, very few are closed, and the bigger ones here, there are some bigger ones occluded in this condition. But as you see in our other pictures, there's also some tumor-related net formation, which is destroying vessels. But basically, these net vessels are more or less okay. But look here, this patient, yeah, this patient, or this, or this, or this, or this patient, they have not a single open vessel in the biopsy we have checked. The size of the biopsy was always the same. Yeah, this patient here, for example, looks close to normal, but the small vessels are more affected. And some of the patients are not, maybe these three here, we should consider not occluded, especially occluded, but this 5 here have really massive problems, and they all died from lung disease. And the future, we will go to adhesions. Adhesions is, I just put in one figure. This is adhesion which is observed after surgical trauma in the belly, and we see clearly here that this is citronylation. This is fully citronylated. There's also fibrinogen included into this. Fibrinogen and nets form together a complex which is very stable because it is resistant to DNase and to protease. It is it could only be digested by the concomitant activity of DNase and protease. But if you add protease, the DNase is gone because the DNase is a protein. So you have always to mix DNase, protease, and then this clot can be digested. That's why this is so stable in the nature. You see the fibrinogen here as well, and you see the hemoglobin. There's a mistake. Yeah, this could not be both fibrinogen. We have to check. And this is without first antibodies, negative control, and this is part 4. Full of part 4, and you see neutrophil elastase, full of neutrophil elastase, and you see on the MPO is more on the surface. It's less stable in the tissue than neutrophil elastase, and we conclude that the vessels and the adhesions form because of net formation, which is then forming this strong structure together with fibrin and fibrinogen. So we are currently first experiments doing with treatment of mice with a DNase 1 like 3, and this mice do not develop the same amount of adhesions, and we are currently planning a study in humans next year. No. Oh, this is just a flow inside. Now we go to a topic which is maybe often ignored by the scientific community. That is net ripening. If nets are not cleared properly in time, they get some process of ripening. And one of these is citronylation. It's not, so of course, citronylation of histone H3 is done during the net formation, but the PAD4 is still active after net formation, and it's planted in the tissue. And there is a tissue citronylation, which is affecting several proteins. I give some examples. One is serpines. The serpines, this serpine here you see, this is a bait, and you see this is the enzyme. No, sorry, this is the protease. The protease is eager to eat the targets, and now the serpine comes and controls the activity, and now the protease cannot anymore cleave. It's controlled with the serpine. Yeah, but next picture shows you this is again the same protein, and you see now this is now citronylated serpine. And citronylated serpine does not bind to the bait region, and that's why it is not blocking the activity of the protease. And one example of what is going on is here. As you see, you have a wound here, a wound formation in the vessel. You see the thrombin is inactivated by the serpine, and that's why there is no formation of a clot. If this is citronylated, the serpine is citronylated, you see that it can cleave the beta and beta peptides from the fibrinogen, and then you see the clot formation in this. Basically, this is a good news. You can get a better healing of the tissue. But of course, as you can imagine, this is only a drawback. If you have intravascular activity, then you see that may also be thrombus formation and the condition of the serpine citronylation, which is basically done by the PAD4, which is planted in the tissue after net formation. And another topic which we are currently addressing is ectopic calcification. You see this mouse here, it's completely full of calcium deposits in the tissue. It's because of fetoin knockout. As in, fetoin is a protein which is preventing the calcification of the tissue. It's binding to the germs of the crystallization process and removes them in the liver and the kidney, and so that you can prevent the tissue calcification. And if this does not work, you look like this. But the question is now how can that be related to net formation? In a normal cellular compartment, you have millimolar of magnesium in the nucleus and in the cytoplasma, and you have millimolar magnesium outside the nucleus, outside the cell. And you have calcium, it's very different. Outside, you have the same millimolar concentration of calcium, but here you have only nanomolar inside the cell. So that means the nucleus, the nucleus of the cell, the DNA is bathed into magnesium. It's a magnesium salt of the DNA, not a calcium salt. And you see, this one is magnesium now here on the DNA. And if the DNA comes now in the vicinity of the cell, outer of the control of the nucleus, it can change the magnesium with the calcium, and now this magnesium calcium exchange may start the process of ectopic calcification. That's our hypothesis, and we also have some data supporting this. If you now look for the size of the phosphate in the backbone, the hypothesis left side is the hypothesis in the textbooks. The calcium can bridge two phosphates, and then it's a calcium salt of the DNA. But we measured the distance between the phosphates and the size of the calcium ion, even hydrated, is not able to bridge these phosphates. The distance is too big. So it means the calcium is not bound in this way. It's just bound in another way. You see here the calcium is too much distant from each other and cannot bind to the so this, you see the hydration here of the calcium. It's a hydrate. And what you see now is the calcium binds in that way. One calcium per phosphate, and another there's one positive charge for the binding to the phosphate, but there's another positive charge left over for further structural additions. And that could be that you can get calcium in this way, and then you can get phosphate, and you get calcium, you get phosphate. And if you look for this, this is basically the start of a hydroxyapatite generation close to the DNA, because as the distance between the phosphate is too big to be bridged by a calcium. So we can only bridge in this way. And that's why the DNA may be a target of calcification. And if you look for the clinical data which are reported since years, there's always some dead cells related to the calcification process. And now let's go for the fibrinogen, which is also related to the net formation, because the citronylation of the fibrinogen, here, if you had two arginines in the thrombin cleavage site, and these are citronylated in the fibrin alpha beta chain, and that means if there's citronylation, you cannot cleave the alpha chain and the beta chain properly, and what the result is a very fragile fibrin clot. And another way, when you have formed the clot, you need the plasmin to lyse the clot, and this arginine here is necessary for the cleavage of the alpha chain, which is cleaved before beta and gamma, and this arginine here is also citronylated, and that means plasmin is not able to digest the alpha chain properly. It is able to digest in the time, but it's not very fast. And we observed that the lysis time was longer for a factor 10. And this is the fibrinogen. This should be before. Here you see the sizes again. Here it's just to remember you. Here you see the fibrinogen alpha beta gamma chain. You see if this is citronylated, yeah, you do not form the well-ordered fibrinogen, which is usually formed in our bodies, but you see that this is disturbed, and this fibrin is not very stable because the cross-linking does not work as well as it should. And distributed fibrin formed by partially citronylated fibrinogen, and if it's fully citronylated, then you cannot even find any form of fibrin formation in this condition. Citronylation of the thrombin cleavage site of fibrinogen prevents bona fide fibrin formation. And now we come to the lysis process. Now we have the lysis. Here we see this is the first cleavage, which is necessary. Then the second cleavage goes on. And after this, we have the three fragments, the D fragment, the D fragment, and the E fragment. And the D, of course, is formed as a dimer. And this cleavage site, now we go here. This one is citronylated here, this one. So that means this and this one, and then it cannot be cleaved by thrombin anymore. Then the whole process is stopped early. So it means the lysis is delayed because of citronylation. Citronylation induced by uncontrolled formation of citrulline due to the PAT4 in the tissue. And this is now, we checked if you do really find these citronylated peptides, and you see here, this is not naive and citronylated fibrin detected in human biopsies. You see here, this is the peptide which is the basis of the alpha chain, the beta chain, and gamma chain. And this is the peptides we detected. The red ones are those which are native. So it means they contain arginine, and the other ones are those which contain citronylated citrulline instead of arginine. And you see it also for alpha, beta, gamma chain. Gamma chain is less affected, but here you see beta and alpha chain are heavily citronylated. And this is a measurement from the tissue. Citronylation of the C-terminal plasmin cleavage site of the fibrin delays fibrinolysis. The question is now, are nets good or bad, guys? It's evolutionary stable net formation. And as it is evolutionary stable, it should have some advantage above those which have lack net formation. We conclude that this is important for the defense in daily life against certain pathogens, especially against fungi and bacteria. However, improper clearance of nets may also fuel chronic inflammation and autoimmunity. That's basically, which is already written in the textbooks. The autoimmunity, how this is fueled, is sometimes not very keen how they describe this. But there is a relation between autoimmunity and continuous inflammation, and that's basically formed from the study of Lars Rönnblom, who is showing that interferon is a major player in systemic autoimmunity in lupus, and that this is driven by interferon, and this could be induced by DNA, which is extranuclear. Aggregated nets orchestrate the resolution of inflammation because they have an anti-inflammatory phenotype. They degrade like crazy cytokines. Yeah, they remove the cytokines in the fluid. You can add aggregated nets. They digest immediately the cytokines, and especially the chemokines. With one exception, IL-8. IL-8 is resistant. However, aggregated nets can occlude vessels and exocrine ducts and persist. Aggregates are candidates to drive ectopic calcification, neoacidification, and adhesions. And with much help from my friends from all countries of the world, and not from all countries, from all continents of the world, and thank you for your attention.
Thank you so much, Martin, for your very stimulating talk. I'm sure there are questions. Please, as I wrote in the chat, you can ask questions in the chat, and we will read them and try to address them. So please don't hesitate to ask questions. It's all to be interactive, and Martin is waiting for your questions, actually. Martin, I can start with the extracellular calcifications. That's really also a clinical problem, unmet need in autoimmune inflammatory disease, in particular in dermatomyositis and also in scleroderma. We don't have anything to do against it. Do you have, but at the same time, they might be very diverse in their pathophysiology. They also they go primarily not to the same regions, actually, which might indicate different pathophysiologies, and they have similarities and differences. Did you have the chance to have material if neutrophils are present or debris is present, and if really net formation, as you were saying, is playing up there?
We are not investigating scleroderma these days, but we plan to do it. And unfortunately, one of our co-workers who is dealing with the issue is now left our university and is moved to Düsseldorf. Maybe you know him, and that's why we were not very clear that we have to look for scleroderma. This was just a new addition in the last days, and but we will keep contact to Jörg and will investigate it. The question is the net formation is a citronylation. The citronylation is my idea, my hypothesis is a culprit of this turnover of the tissue, because citronylation is a very stable stuff. It cannot be reverted back to arginine. It's not thermodynamically stable, but it can only be replaced by new proteins, by new structures. And as you know, the structure is in a condition of inflammation. New replacement of structures of tissues is not so frequent and not so effective. So I guess the citronylation is basically a marker of that. Here was inflammation which is already been cleared, and it takes time. And if you extended fields of inflammation, you may have more problems to reconstitute the tissue in a proper way, and that's why the system just starts with doing fibrosis, and that's my hypothesis. And of course, calcification is not the only way to calcify is DNA, but DNA is one possibility, and we do this now in vitro. In vitro, we observe that the DNA together with the fibrinogen and fibrin formed from fibrinogen is responsible for a certain kind of calcification. But this is in the beginning of the experiments, and we cannot really attribute it to certain diseases now. The fibrin net axis we only started working on with the net in the adhesions. And we will, of course, continue working with other diseases which are culprits, which are candidates for this calcification. Yeah.
Wonderful. I think this would really address a very high unmet need in several inflammatory diseases. And ectopic calcification is really problematic for the patient.
Especially the skin is sometimes affected even more than you expect. When you look for the X-ray, you don't not see it very well. Yeah, but if you go for microscope, you see sometimes in the hair follicles you see calcifications, and this is not only in the hair follicles, but there it is most easily detected. But these microcrystals are very hard to detect, and they are still inducing inflammation, because as you have seen, the nanodiamonds are just 10 nanometers in size. This can induce inflammation, and of course the nanodiamonds never disappear, because the net's formed, but the diamond is still diamond, and it cannot be removed, because phagocytes, when they eat it, they also get a shock. And spit it out again. So that means there are some hydrophobic stuff, which is really a problem in connection with the neutrophils.
Very interesting. So we might get you in contact with some juvenile dermatomyositis experts, because there you have really plates of calcinosis sometimes. That's very disturbing.
Yeah, we would be very happy to investigate it. Yeah.
Wonderful. Caroline has a question. Maybe Caroline, you can switch on your mic because you're a co-host and ask your question.
Yes, actually, can you hear me? So now I'm co-host, so I can interfere in. So thanks a lot.
I can hear you. Wonderful. Yeah.
Very good. Thanks a lot. As usual, you showed very impressive pictures and stainings of the nets. And my question was, seeing all these net clogs, so do you think this is just something that happens in everybody in a certain condition? Or do you think, like we have for the coagulation, that some people are more prone like to bruise this and do overnetting, or have a lack of resolution, and this is why maybe some individuals have more than others?
I'm absolutely convinced that there is a difference in the people, susceptibility to net occlusions, to net formation, to net problems. As you can see, some of the patients which have a infection with corona, they have nothing. Everything is fine. If you have a risk factor is a vasculitis, if you have already vasculitis going on, the vessels are kind of rough inside, and these rough areas, which are basically like scars, they are eagerly taking up these nets, and then they occlude. And when they occlude, then of course you have the problem is not the net formation. The problem is the occlusion. And of course, if you have DNase 1 and 1-like-3, you usually have a good condition, but on the long run, it's a time of age. Then you may also have some areas where you do not get it controlled. There's some data from a family of Saudi Arabia which have no DNase 1-like-3. In mice, it doesn't matter. In mice, it's enough to have one of the both DNases. But the family in Saudi Arabia has no 1-like-3, and this family has all these members have the lupus, systemic lupus, a strong lupus. So that means the clearance of the lupus antigen, whatever it is also requires DNase activity. It requires DNase 1-like-3, because 1-like-3 is better cleaving chromatin, whereas DNase 1, as you remember from the footprint, DNase footprint is only cleaving naked DNA. So that means the DNase 1, which is still present in that family, is not sufficient to remove the DNA which is generated inside the body and usually metabolized. Nobody notices it, but this patient cannot handle it, and then they get lupus. And lupus is also, of course, driven with interferon is included, and there are so many conditions. But of course, every one of us has nets also in the circulation. Every one of us has a coagulation going on day by day in our vessels. Even if we are healthy and young and whatever, it says if you look for D-dimers, there is not zero D-dimer in a healthy young population, but there is little. and this can be controlled, and the vascular occlusion with the thrombus is avoided because the Lupus system is working well. But of course, the Lupus system may have problems, and then you can also get genetic susceptibility to the net formation and net occlusion, and all the net-related diseases.
And I can imagine that evolutionary, under some circumstances, this could have been even beneficial, to have, because as you said, the neutrophils are also the good. So maybe in some conditions, this might have been actually an advantage to have more.
Yeah, it sounds crazy, but in former times, very former times, if you have some wound, it is you got often sepsis in the body. But if you were able to occlude your vessels, and the vessels are not anymore connected to the vasculature of the body, you lose maybe a finger, you lose something, but you still survive because you block the blood flow back into the body and just lose your finger instead of losing your life. But this is one hypothesis which I always discuss. This, I guess, is also seen in a very old Xenopus. No, not Xenopus. In this, it is crab. I don't remember the name. Where the hemolymph, is when some bacteria come in, they immediately clot together, and then the clot is eaten by the macrophages, and this, of course, the clotting of the vessel is in our life is a disaster. But it can also be evolutionary. It can also be an advantage to be able to clot parts of the body to just lose a part of your body and not all the body. That's basically what I always remember, because of the evolutionary stability of this process is so amazing. So that I could not understand that it has no only disadvantage. It cannot be.
Thanks a lot.
So maybe I have a last quick question, Martin. When I looked at your DNase knockout mice where the vessel occlusion was occurring, my understanding it's really due to net formation, right? This is net formation. My question is there secondary vascular activation, endothelial cell activation, or even vessel wall inflammation, or is there maybe even primary endothelial cell activation and vessel wall, also vascular smooth muscle cells activation and proliferation? Because what you are showing reminds me a lot of what we see in Buerger's disease, or what we also see in vascular inflammatory models, like in pulmonary arterial hypertension, actually. Did you check whether this is then really a primary net formation, or is the net formation supported by endothelial cell activation?
I'm sure that the immune system is usually has not only one actor. And what we observe is, I can only describe that the mice die very fast, and that basically the only thing we can find in that corpse, which we collect or which we euthanize, is a vascular occlusion, which we can clearly see in several tissues. We also see it. We see it in liver. We see it in kidney. We see it in the lung, and maybe also skin. I'm not sure. I don't think we have looked for skin. Yeah, but we see this vascular occlusion, but we cannot see anything else. But the death is very fast. It's some days. Yeah, and the neutrophils go up. Of course, they have to propagate. That means it takes approximately 1 day to get enough neutrophils to get neutrophilia, and then even 2 days later they are already dead. That means it's really a strong hint that the initial activity is a net formation.
So that's quite similar to Buerger's disease, where you have no vessel wall inflammation activation at all. But there's a thrombus, and so there is obviously thrombin involved, which you didn't have. But maybe again, worthwhile to check maybe in Buerger disease samples whether you have net formation there. Maybe.
Yeah. Do you have samples?
This is a typical angiology topic. So I think you need to contact angiologists for this. We only see them for differential diagnosis. Actually, these are the male young smokers that get vessel occlusion and severe necrosis. Actually.
Yeah, but you know, as a physician, there are several conditions in the body where you say, yes, we tried already what we can do with anticoagulation, and it did not work. Yeah. And then you get consumption of the and then you get a depletion of the effectors, and then everything is destroyed. But the initial sentence is usually, we did already. Yes, you have to no, we did already, but we did what we can. I think there is something which we have to do, which is basically not only going for the classical canonical anticoagulation, but for new ideas, maybe by DNase 1 like 3. That's what we are currently doing, at least for some diseases. And the disease, of course, is COVID in these days and lupus.
Wonderful. Thank you, Martin, again, for the wonderful presentation, stimulating presentation as always, and also a wonderful discussion. Thank you so much, Martin. And please stay online because now we have Mariana Kaplan.
I do. I just switch off my micro.
Wonderful. And Caroline introduces Mariana.
Yes, I just say a few words because Mariana is really also a big figure in the granulocyte field. She's chief of the systemic autoimmunity branch and a deputy scientific director of the National Institute of Arthritis and Musculoskeletal and Skin Diseases in Bethesda. And she did a lot of work with characterizing granulocytes, neutrophils, subtypes, identifying the beasts, as Martin would say, and how they actually activate the immune system. So thanks a lot, Mariana, from a different time zone to join, and we are very much looking forward to your talk.
Well, thank you so much, Caroline and Oliver. And again, my apologies for the technical difficulties. You would think that by now I should be a pro, but the computer crashed last minute and I had to figure out what to do. Anyway, I'm delighted to be here and I'm very excited to participate. And thank you to Martin too for an outstanding presentation. So my talk will be, I think, complementary to Martin's, and I hope I won't be too repetitive in some of the aspects, but I basically hope to introduce some concepts that I think are relevant for how neutrophils may be crucial in autoimmunity. So first, I'm going to talk about some of our recent work on sex differences in neutrophil biology. I will discuss some aspects of neutrophil heterogeneity and link that to how this may lead to dysregulation in autoimmunity and vascular damage related to autoimmunity, particularly in the context of NETs. How we may target this neutrophil dysregulation, and at the end I'll have a few thoughts on some recent work we published, mostly on pediatric COVID and NETs, and something related to different variants of COVID, of the virus and the induction of NETs. So as you know, for all of us studying autoimmunity, these are complex heterogeneous diseases where we now see them as multi-step processes where both genetic and environmental factors play a key role in inducing a susceptibility phase in individuals that we think develop profound innate and adaptive immune dysregulation early on. Of course, it's hard to know how early, but for different diseases such as lupus and RA, we know that innate and adaptive immune dysregulation may precede clinical disease by many years. And eventually, through a variety of hits from the environment and poorly understood stochastic events, we see in many of these diseases the subclinical development of autoantibodies that target both cellular and extracellular autoantigens. And then eventually, again, through another hit and bad luck, individuals predisposed a percentage of individuals predisposed to develop the disease will develop first the acute manifestations. In many of these conditions, periods of flare and remission, either spontaneously or triggered by medications. And as our patients live longer and we have done a better job at controlling some of the acute manifestations, we see more and more the chronic complications that lead to significant morbidity and mortality. And my group has been particularly interested in understanding why patients with systemic autoimmunity, particularly lupus, have such accelerated rates of premature cardiovascular disease. So one key feature, as you know, in systemic autoimmunity is the striking female predominance, where many of these diseases, both organ-specific, but especially in systemic autoimmunity, have a stark predilection to affect women. And of course, this is complex, and it's sex chromosome versus hormonal factors, and there is likely a convergence of many different variables that may lead to a higher prevalence of autoimmunity in women, and differences in innate and adaptive immune responses have been reported between females and males in healthy conditions. And of course, in lupus, the prevalence is particularly striking, that for every man with lupus, there may be about 10 women that are affected. So most of my talk will focus on lupus, and for this audience, I don't need to say much about the disease, but what I would like to emphasize, of course, is that the sine qua non of lupus is the formation of antibodies that target nucleic acids, DNA and RNA, or proteins binding to nucleic acids, which is histone. So these are main autoantigens in lupus. Of course, the dysregulation of the type 1 interferon pathways that is observed in a significant proportion of lupus patients at some point during their disease. And again, this striking increase in the prevalence of accelerated vasculopathy and atherosclerosis in women that otherwise may not have other traditional Framingham risk factors. And so why is lupus more prevalent in women has been the focus of intense research. And of course, this is complex, and is the interaction of, we believe, genetic, epigenetic, and environmental mechanisms. Some poorly understood, for example, the role of the microbiota, but that's an area of, well, you know, where there's a lot of intense interest in diabetes and in other conditions. We're trying to explain how this may affect differently autoimmune responses in women and men. And so, as Martin mentioned, you know, autoantigen generation is a key phenomenon. Responses to aberrant autoantigen generation, it's a key phenomenon that characterizes autoimmune diseases. And for many years, the dogma was that the main mechanism or the main dysregulation occurred in apoptosis, where you had increased apoptosis and or impaired clearance of apoptotic material, and that in predisposed hosts, this would fuel the fire and contribute to induction of type 1 interferon responses and the dysregulation of both innate and adaptive immunity. However, this view was oversimplified, and understandably so, because until way more recently, we knew much less about other types of cell death. And now we know that cells can die in many other different ways. And also, it is not only how cells die, but also which cell dies, where it dies, what is the surrounding microenvironment of this dying cell, that may be key in establishing whether the response to the cell death is immunogenic or tolerogenic. And in that regard, several groups, including ours, have been interested in understanding how neutrophils live and die, because, you know, as Martin mentioned, this is the most prevalent immune cell in humans, and these cells are constantly being produced by the bone marrow and constantly dying. They don't have a long half-life. But I also want to emphasize, as part of this symposium, that the field of neutrophil biology has, although it really lacked compared to what we know about other cell types, in part because it is technically so challenging to study neutrophils for many different reasons. The advances over the last decade have really changed how we view neutrophils, and I believe place them in a central role on how they may shape the immunologic landscape in health and disease. And besides well-established roles of neutrophils in antimicrobial responses, we now know that they are crucial in the coagulation cascade, that they may have certain immunosuppressive roles, at least some subset of these cells. And of course, in the context of autoimmunity, we have been particularly interested in understanding first how differences in neutrophil cell death, particularly through net formation, may be crucial in the initiation and perpetuation of autoimmunity, but also in some of the end organ complications, but also this concept of plasticity or cellular heterogeneity that is still fairly poorly understood, but that it is gaining traction, and so that we don't think of neutrophils as this monolithic kind of boring cell type that is just there to sense microbes, go there, kill the microbes, and then die, and rather to think that there are different stages or subsets, that's still a little unclear, of neutrophils that may have differential roles during homeostasis and disease. And I would like to emphasize some key, really interesting aspects that have fairly recently been highlighted in neutrophil biology. One, especially from Andrés Hidalgo's group in Spain, is that neutrophils have their own circadian rhythm and internal clock, and that a neutrophil in the morning will look very different than a neutrophil at night. So when we study neutrophils, when we harvest them, may be key in what we will find. And this is something that we really need to take into consideration. And not to forget that mice and humans have very different circadian rhythms. I mean, mice are nocturnal, we are usually not. And so the findings we may see may be quite different if we study a mouse neutrophil in the morning and a human neutrophil in the morning. So just something to keep in mind. Of course, the role of the microbiome in how it modulates neutrophil biology is still an area that is quite unclear and certainly probably quite important. I will talk a bit about the role of sex hormones in neutrophil biology. We know very little of how the life cycle of a human may affect or the transition of different stages of neutrophil phenotype and function. The role of genetics remains to be better characterized. And of course, the role of the tissue microenvironment and how neutrophils interact with different stromal cells in different tissues may be quite relevant on what type of response is induced at the specific level of that tissue. And so I won't spend a lot of time. Lucky, I deleted my slide on what NETs are because Martin gave a beautiful explanation about them. But we have been very interested in the context of neutrophils in understanding whether NET dysregulation is an important mechanism of autoantigen generation and the induction of organ damage. And so we now know that in addition to microbes, there are a variety of sterile inflammatory stimuli that have the ability to induce NETs in predisposed individuals. And so immune complexes, a variety of autoantibodies, but interestingly, not every autoantibody, various crystals such as uric acid, but also cholesterol crystals, complement components, and platelets. And the interesting thing is that NETs do carry many of the autoantigens that are known to be key targets of the immune system in autoimmune diseases. So not only double-stranded DNA and histones in the case of lupus, but they have high levels of MPO and PR3 if you think about ANCA vasculitis. And because of the citrulination of certain molecules such as vimentin and enolase that are present in the NET cargo, they may represent important sources of citrulinated autoantigens. And we think that this concept of impaired NET degradation, increased NET formation, is shared in a variety of autoimmune diseases, not only lupus, but it has been demonstrated, not only the ones mentioned here, but for example, in antiphospholipid syndrome and in idiopathic inflammatory myopathies. And so is this operational in vivo? We believe that's the case because when we have looked at various tissues, and other groups have also found similar things, we do see that tissues affected in these diseases are infiltrated by netting neutrophils. So this is not just a phenomenon of that harvesting neutrophils and looking at them in vitro, but that there is evidence that this is occurring in vivo. And so what I was alluding to is that it is interesting that our group and others have found that certain autoantibodies, but not all, or certain immune complexes, but not all, have the ability to induce NET formation and this is characteristic in certain diseases. So for example, we have found that lupus RMP immune complexes are strong inducers of NET formation, yet other type of autoantibodies, not necessarily so. In rheumatoid arthritis, for example, antibodies to citrulinated vimentin appear to be very good at inducing NET formation. And in the case of myositis-specific antibodies, we found a couple of years ago that anti-MDA5 antibodies were particularly striking at inducing NETs, but for example, anti-Jo1 weren't. And why is that? We're still not sure. It may be something related not only to the specific autoantibody, but the quality of that IgG or other factors that we are still poorly understood. It's something we think a lot about, or whether there's something related to the genetic makeup of individuals who make those antibodies that may make them more prone to form NETs. I don't have an answer to that. And if someone has some ideas, I would love to discuss that with you. So just to summarize, the idea here is that NETs generated in autoimmune diseases, and particularly NETs generated by this subset I will discuss, the low-density granulocytes, are particularly good at inducing innate and adaptive immune dysregulation. Among them, for example, the ability of NETs to activate the NLRP3 inflammasome machinery and increase the production of IL-1 and IL-18, thereby propagating inflammatory responses in tissues. The ability, as Martin mentioned, to induce type 1 interferon by plasmacytoid dendritic cells and also by certain myeloid cells. The ability to activate adaptive immunity, both T cell and B cell responses. And importantly, NETs, especially NETs generated by certain neutrophil subsets, appear to be quite vasculopathic, both by their ability to kill endothelial cells through distinct cell death mechanisms that get activated in these cells, but also through the activation of coagulation, both by activating coagulation factors, inactivating anticoagulants, and activating platelets. And so the list really has expanded, and NETs have been implicated in a significant number of both systemic autoimmune diseases, but also autoinflammatory syndromes and some monogenic autoinflammatory syndromes that we have had the opportunity to study at the NIH in collaboration with Dan Kastner and with Peter Grayson, and recently with Bexas. We see evidence of NET dysregulation in association with vasculopathy. And as Martin mentioned, there has also been, an explosion of data implicating NETs in a variety of metabolic syndromes, in cancer, in gallbladder stones from work from Martin, and more recently in COVID-19. And one aspect of NETs that is interesting is that they may serve as messengers that carry certain molecules that may activate target cells. And among them, for example, it's not only the presence of citrullinated molecules. I won't have time to talk about some recent data we have and current under review, but other post-translational modifications in proteins, such as carbamylated proteins, appear to also have a role in target cells. And there is a significant amount of carbamylation of proteins in NETs. NETs carry also a variety of small RNAs, and we have found that these have the ability to, for example, activate endothelial cells. And I think the field continues to be expanded. There is such an abundant cargo of proteins that are modified that we need to better understand how they interact with target cells. But the other important aspect, going back to the DNA and the RNA, is that something characteristic of NET formation that may not be seen in other types of cell death is that the nucleic acids become oxidized during NET formation. And this may not necessarily be the case in other types of cell death. And this nucleic acid oxidation, we believe, is key because these oxidized nucleic acids are less likely to be degraded by both intracellular and extracellular sources, nucleases, and therefore more likely when internalized to be sensed, for example, by the cGAS STING pathway and lead to the production of interferon. So I do believe, based on our work and the work of others, that NET generation may be particularly immunogenic when you compare with other types of cell death, and may be a significant mechanism that may lead to responses to these modified autoantigens in individuals at risk. And of course, time will tell whether that's a predominant source of these autoantigens or whether other mechanisms play stronger roles. And of course, now there is also evidence that certain genetic polymorphisms may be associated not only to the development of autoimmunity and SLE, but that by themselves, they also may lead to enhanced ability to form NETs. And this has been reported by different groups. And for many, you will recognize here several of the gene associations with SLE. And interestingly, for example, work from Betsy Barnes showed a couple of years ago that individuals with susceptibility gene, who carry the IRF5 susceptibility gene, even when they haven't developed lupus, if you study healthy people who carry the risk allele, they will make more nets compared to people who don't carry this risk allele. And we found something similar in the case of STAT4. So this may tell us, of course, it's a chicken and egg question. And one would say, well, if you study SLE, they're already forming nets for other reasons. But the fact that people who haven't developed SLE and carry these risk alleles tend to develop more nets, I think supports the possibility that net formation is an early event that may lead to modified autoantigen generation and inflammation and be one of the factors that could promote autoimmune responses. So going now to neutrophils and sex differences. This was an area that when we got involved was actually quite understudied, even if differences in the morphology of neutrophils between females and males had been described many decades ago. And if you look at all literature, it's interesting that you will find evidence that there are differences in neutrophil cell numbers between females and males, that the morphology is different. Of course, there is the presence of the drumstick in the female neutrophils, and also that there are some fluctuations during the menstrual cycle in the number of neutrophils, and therefore all studies had already implicated estrogen and progesterone or sex hormones in neutrophil functionality. But there was very little really that we could tell when we started studying or asking this question. And this came in part from a collaboration with Mark Davis' group at Stanford and David Herrmann, also from Stanford, who contacted us because when they had done a transcriptomic analysis of peripheral blood of healthy individuals and look at existing databases, what was clear when you compare in young adults, males and females, healthy people, was that the main difference in the gene expression was in genes related to neutrophil biology. And long story short, what we found here was that male neutrophils are more immature than female neutrophils in healthy young adults. And this translated to the finding, and this was replicated in different cohorts we looked at. And this difference in maturation translated into the fact that female neutrophils in healthy people are more activated than male neutrophils. And as part of that process, we found that female neutrophils tend to form more nets, either unstimulated, stimulated with microbial stimuli, or with sterile inflammatory stimuli, for example, such as lupus serum containing autoantibodies and immune complexes. Suggesting that even in healthy females, there may be a higher predisposition to generate autoantigens. And you would think that this may be one driving factor that could potentiate, when you have the wrong genetic makeup, the induction of autoimmune responses. And so what suggested that this may be hormonal is that when we looked at in pregnancy in healthy people, during pregnancy, the female neutrophil acquire more the phenotype of a male neutrophil. And then after the delivery, during the postpartum, then the neutrophil phenotype went back to how it looked prior to pregnancy. So rather than being something chromosomal related, we thought this suggested that there are hormonal factors, as may happen in pregnancy, that may be implicated in the differences in maturation status and activation of these cells. And so we went on and then did our own studies where we tried to do more comparative studies between female and male neutrophils. And this is all healthy adults. And what we saw is that when we did seahorse analysis and compare female and male neutrophil responses, because the male neutrophils are more immature, they tend to rely more on mitochondrial respiration, because as neutrophils mature, they tend to lose mitochondria, but more immature neutrophils have more mitochondria. So male neutrophils had a higher mitochondrial activity. They had higher mitochondrial DNA content. And interestingly, when we added estradiol to the male neutrophils, they acquire the bioenergetic capabilities or the bioenergenic profile of the female neutrophils, suggesting that hormones were regulating immunometabolism in neutrophils from these individuals. And then we went ahead and purified male and female neutrophils from healthy adults, young adults, and look at differences in transcriptional analysis. And the most striking finding was that the main genes that were differentially expressed between females and males were interferon-regulated genes. And so that females upregulated a variety of interferon-regulated genes in a very consistent manner. We replicated this in different cohorts, and it's something we keep seeing on and on. And so, and I'm showing you here, if you look at an interferon score, females have a significantly higher interferon score. Interestingly, this is very distinct to neutrophils, because when we have done single-cell RNA sequencing analysis and compared female and male immune cell subsets, we do not see these differences in the interferon-regulated genes in T cells, in B cells, in NK cells, etc. It is very distinct to the neutrophils. And so we have been asking why, and we looked at whether female neutrophils express different levels of the type 1 interferon receptor, and they don't. We looked at whether the females had higher levels of circulating interferons, and they don't. It would appear that the machinery, the downstream machinery that responds to interferons is significantly more activated in the female neutrophils than in the male neutrophils. And we think this is really related to the differences in maturation status, that the JAK-STAT signaling pathway is another pathways that may be associated with type 1 interferon responses are upregulated in the female neutrophils compared to the male neutrophils. The implications of this remain to be seen, but of course, this could be quite crucial in the context of diseases where type 1 interferon or response to type 1 interferons plays key roles, such as in autoimmune responses. We also wonder about the implications for COVID-19, since, as you know, males have, at least for the initial strains, tended to do significantly worse, and whether this has to do with response to interferons is something that we have speculated as well. And so to try to dissect whether this is hormonal or chromosomal, this is tricky because it is hard to manipulate neutrophils in vitro with hormones because they're terminally differentiated, and we have tried all sorts of things. So we did it a bit in an indirect way. We got blood and neutrophils from patients with Klinefelter syndrome. So as you know, these patients have an extra X chromosome, so they are males, but they have an XXY karyotype. And so we thought, if this is a gene dosage effect, an X chromosome dosage effect, then we would expect that the Klinefelter neutrophils would behave more like the female neutrophils, be more mature, and respond more to type 1 interferons. But in fact, the Klinefelter males and the males without Klinefelter, their neutrophils behave very similarly. Furthermore, when we studied children before puberty, so when we would think the levels of sex hormones may be more or less comparable, we did not find any differences between girls and boys in the induction of type 1 interferon genes or in the level of maturation. So although indirectly this suggested to us, and with all the other previous evidence I mentioned, that this may be more related to effects of sex hormones. And we are currently very interested in trying to investigate how sex hormones modulate neutrophil maturation and perhaps other responses that may be quite relevant on how immune responses could be, or in chronic inflammatory responses could be manipulated beyond immunosuppression if you think about hormonal manipulation very early on, something we are just starting to think about. And so we have really been interested in understanding this concept of neutrophil heterogeneity, both in health and in disease. And it's been very hard to do this because, as you know, single-cell RNA sequencing analysis in neutrophils has been difficult, much more difficult than for other cells. But through the development of different pipelines, we initially started with whole blood and found that indeed, in healthy males and females, not all neutrophils are the same at the transcriptional level. And we identified at least what appeared to be 4 types of neutrophils by transcriptional analysis in peripheral blood in healthy individuals. And only one of these subsets is the one that expresses high levels of type 1 interferon regulated genes. And it is not that females have more of this subset, is that within this subset, they respond more to type 1 interferons. They have higher levels of type 1 interferon regulated genes than the same subset in males. Suggesting again this hyper responsiveness of this subset of female neutrophils compared to male neutrophils. And we're trying to characterize what this really means. We recently published a modified pipeline by which in purified neutrophils, not using whole blood, but just in purified neutrophils, we have been able to successfully do single-cell RNA sequencing analysis. This was just published a few weeks ago. And through this modified protocol, we have again confirmed that in healthy adults, there are at least four types of neutrophils described at the transcriptional level. And again, when we have done very detailed analysis, we have seen that there are different types of genes that they tend to upregulate. And some of them overlap, but there are some that are quite distinct for these groups. And there is only one of them that expresses high levels of type I interferon-regulated genes. And we have confirmed this by using single-cell Western blot, where there is always a small proportion of the neutrophils that also at the protein level tend to express high levels of type I interferon-regulated proteins. So what is interesting, and I want to emphasize, is that if we were to look at this subset by cell surface marker, and here we did site-seq, there's no difference in at least many of the markers we have looked at for these different subsets. So if you were to try to, at this point, what we're really focusing on is, are there distinct markers, cell surface markers for these subsets that may allow us to purify them and look at functionality? We are not there yet, because at least with the initial markers we did, there was no way for us to differentiate them, only that by transcriptional analysis. And when we have done trajectory analysis in this subset, what it would appear is that one of the subsets that is circulating is kind of immature. And from there, you will get the generation of a more mature subset, which is the NH1, which is what we would characterize as the typical mature neutrophil. And then they then diverge into two lines as terminally differentiated cells. One, this NH2, which tends to be more of a low transcriptional subset that, you know, has a much lower transcriptional signature than the other types. And then the other terminally differentiated neutrophil is the type I interferon responsive subset. So this is where we are right now. We have been trying to characterize specific transcription factors that are associated with each type of neutrophil subset. But what we really, really would like to do is understand what these subsets do in health and disease. And what is this type I interferon responsive subset meaning? Is it a cell that has patrolled and has sensed danger? Is it a senescent cell? We don't know yet, but we're hoping to have more answers in the future as we try to purify them and do functional analysis. And so moving to heterogeneity now in the context of autoimmunity, as Martin mentioned, we have been very interested in this subset of neutrophils that we characterized over a decade ago. And we started being interested in it because when we were doing Ficol gradient, trying to purify mononuclear cells in lupus, we always found this contaminating neutrophil subset where neutrophils shouldn't be because they usually have higher density and they go to the bottom. But in the case of lupus, we were always finding, in some cases, many of these neutrophils in the mononuclear cell layer. We call them low density neutrophils because they had to have lower density to be able to be found there. And we spent a lot of time trying to make sure these cells didn't represent just an epiphenomenon or were innocent bystanders. And what we believe now, and I think what other groups have also found, is that these cells, in the context of autoimmunity, represent a pro-inflammatory subset, and that with distinct functions or pathogenic activities, including their very enhanced ability to spontaneously form nets ex vivo. So there's nothing we can do to further stimulate these cells to form nets. Once they are out of the blood, they already come out of the circulation, really max out in their ability to form nets. And these nets are endowed with a significant ability to induce vascular damage. We think this is something quite characteristic of these low-density granulocytes, that they are particularly good at damaging the endothelium, and a shared feature of these cells in the different diseases that tend to have a higher prevalence of them. And so this is a list of some of the conditions where they have been described: lupus, ANCA vasculitis. We describe them in some types of drug-induced vasculitis, in psoriasis, inflammatory myopathies, metaphospholipid, and also recently in some autoinflammatory syndromes such as PAPA and DADA2. And some of the shared features, of course, of these diseases is the enhanced vasculopathy or vascular damage. And we think this may be a unifying factor that may promote this blood vessel damage in these conditions, and it's something that we are actively studying. And interestingly, these cells appear to be transcriptionally quite different from even normal density neutrophils from the same patient. So autologous normal density neutrophils look very much like control neutrophils, but when you look at bulk RNA sequencing of the low density granulocytes, they have a distinct gene signature. So transcriptionally, they are different. When we have done single cell RNA sequencing analysis in mononuclear cells, and we found these low density granulocytes, they seem to, in lupus, be responsible for the most striking type 1 interferon gene signature when compared to the mononuclear cells. So they may be strong contributors to the characteristic type 1 interferon gene signature. And they represent some sort of heterogeneous subset where the majority are really mature endothelial neutrophils, and so only a small number of these cells represent bona fide immature neutrophils, contrary to what was originally thought, that this was some sort of emergency granulopoiesis response. We do not think that's the case. We think these are, mature pro-inflammatory cells, different from the suppressor neutrophils described in cancer. We have not found suppressing ability of these cells when you look at a variety of assays, contrary to what has been found in cancer. So even if they may be found also in the peripheral mononuclear layer, they behave very different from the LGGs from cancer patients. And interestingly also, they don't only carry a different transcriptome, but when you look at the proteome of these low density granulocytes, it is different compared to neutrophils from the same patient that are normal density or healthy control neutrophils. And one key aspect is that some of the proteins that are differentially regulated in low density granulocytes are proteins associated with biomechanical properties of the cells. So how cells move in circulation, how they adapt to different sizes of blood vessels, for example, quite different, the proteome in the low density granulocytes. And what we actually found is that we did this, developed this 3D mimetic, where basically we were trying to recapitulate in, not in the best way, but as we could, how neutrophils would travel in small blood vessels, right? And so we thought, well, if there are differences in biomechanical properties, we may see differences in how quickly these neutrophils can travel through this mimetic and exit. And in fact, the low density granulocytes did a much poorer job at traveling this mimetic. They took much longer and they got trapped much more easily than normal density neutrophils from the same patients or healthy control neutrophils, suggesting that these changes in the proteome may actually affect their ability to travel in blood vessels and may get trapped there and may actually occlude blood vessels, similar to what Martin described in the context of NETs. Not so much because they adhere to endothelium differently, we didn't find that, but it's really because of differences in their biomechanical properties, perhaps secondary to differences in proteome that we're trying to better understand. So this may be an additional pathogenic mechanism of how these cells cause tissue damage beyond NETs. And what is the implications for clinical disease in lupus? So as you know, lupus patients, as I mentioned, carry a higher risk for vascular disease. We have been looking at this beyond some of the ways that people look at and doing, for example, FDG PET-CT at the clinical center, where we have seen that lupus patients have a lot of vascular wall inflammation, even when they haven't developed ovascular disease. And this associates with what we call significant uncalcified plaque burden in lupus patients, where you see these irregularities in their coronaries that are not seen in healthy individuals. And our hypothesis has been that lupus represents the extreme phenotype of innate immune dysregulation at the level of the plaque, where activation of the type 1 interferon pathway and aberrant neutrophil biology and net formation may converge at the level of the artery, promote plaque formation, promote unstable plaque and thrombosis enhancement, and that this may be one important mechanism, kind of this thing by which lupus patients may develop accelerated vascular damage. And in fact, when we have looked at a cohort of well-characterized lupus patients at the NIH Clinical Center, and we have looked at many variables that associate with non-calcified plaque burden or with vascular inflammation, we have found that in multivariate analysis adjusting for a number of confounders, the numbers of these low-density granulocytes significantly associate with higher levels of non-calcified plaque. And when we have done RNA sequencing analysis and looking at the LGG signature in patients that have high plaque or high vascular inflammation compared to those lupus patients that look more like healthy people, we see that those with more severe vascular damage are the ones that have a higher low-density granulocyte signature. So although this is just association, it does support the concept that these cells may have important vasculopathic roles. And we're trying to follow these patients longitudinally to assess how variations in low-density granulocyte numbers may associate with progression to vascular disease and how we ideally may modulate that risk. And so how may we target aberrant net and neutrophil dysregulation in SLE? So one of the things we recently published in collaboration with AstraZeneca was we were able to obtain serum samples a posteriori. So we really didn't have cells during the study. We partnered with them after the trial was over. But this was a anti-type 1 interferon receptor trial, and anifrolumab, which, as you know, is a drug that in the US has been approved for lupus. And the idea here is that interferon may be serving as a priming factor for net induction in lupus, and that blocking type 1 interferon pathways could have a beneficial role in net dysregulation and vasculopathy. So because we could only have serum samples, we had to get creative or what we could look at, but we were able to obtain samples from at baseline and also 1 year post, 1 year having had exposure to anti-type 1 interferon therapy. And what we found interesting is that when we look at circulating nets in lupus patients, those patients that had taken the anti-interferon receptor therapy had significant decreases in net complexes, irrespective of whether their disease got better. Their net complexes did go down, whereas those that were in placebo, in fact, the net levels of nets were up. So it did appear that modulating type 1 interferon responses could have an effect on circulating net levels. And the other thing we found that I'm not sure here is that we are actually able to modulate other markers of cardiometabolic disease, such as HDL oxidation and other parameters that have been associated with poor risk, cardiovascular risk in lupus. So suggesting that this should be taken into consideration. And we are in the process of initiating a trial of this drug to see if we can modulate actually vascular function and other more direct vascular parameters in lupus patients. And the other approach we follow was to assess whether blocking the JAK-STAT pathway could be a mechanism to affect neutrophil dysregulation in SLE. So as you know, the JAK-STAT pathway is a fundamental pathway in that have many cytokine signal. And on one hand, of course, type 1 interferons signal through the JAK-STAT pathway, but also neutrophils have a very active JAK-STAT signaling pathway where many cytokines crucial to neutrophil development and biology in general use this pathway for their effects. So we had previously done studies in mice, in lupus-prone mice, where we have found that the administration of tofacitinib in lupus-prone mice led to not only significant decreases in clinical parameters of disease in the mice, but also significant decreases in net formation and vasculopathy, both ex vivo and in vivo. And so we then proceeded to perform a trial that we published some months ago. It was a proof of concept small trial with 30 patients where we gave them either tofacitinib or placebo. It was more of a safety type of study where we wanted to assess if lupus patients would tolerate the drug well. So they got the drug for about 2 months, and then we studied them up to a month later after they stopped the drug to try to assess what was happening. So the drug was well tolerated. It wasn't, the trial wasn't designed to look at efficacy because it was small and short term, but we used it to do some mechanistic studies to assess how would the drug affect certain innate and adaptive immune parameters. And what was interesting and what I want to highlight here is that taking the drug, patients that did not take tofacitinib had actually an increase in the numbers of low-density granulocytes during the trial, whereas those who took the drug had a decrease in the numbers. So this was statistically significant, and whereas total neutrophil numbers did not significantly change. So somewhat suggesting a selective effect of these inhibitors on low-density granulocytes compared to globally on neutrophils. And interestingly, we use an approach here where we randomize the lupus patients according to whether they had or didn't have the STAT4 risk allele, thinking that they may respond different to the drug. And in fact, they did. Those that had the STAT4 risk allele seemed to be more responsive to the drug. And we found not only that patients with this allele, as I mentioned, had higher levels of NETs at baseline, but that those were the ones that when they received tofacitinib, but not placebo, had significant decreases in their circulating levels of NETs, suggesting that short-term use of tofacitinib may actually have a positive effect on neutrophil dysregulation in lupus. And of course, longer duration studies in larger cohorts will be needed to confirm that. But this was also associated, not surprisingly, with significant decreases in the type 1 interferon gene signature, improvements in lipoprotein function, and very importantly, when we look at readouts of vascular dysfunction, in this case, vascular stiffness, a measure of vascular aging, we found that those patients that were on placebo during the trial actually had increases in their vascular stiffness, but those that weren't tofacitinib decreased their vascular stiffness, perhaps related to the decreases of both type 1 interferon responses and neutrophil dysregulation. So of course, there is this other dark aspect of these inhibitors on potentially the promotion of thrombosis that are of concern. So we'll have, with more studies, have to balance and really assess what is the role of these compounds in the context of vasculopathy in patients at risk, such as in lupus. And I want to end here by telling you a little bit about a study we just because this is COVID, and we have been very interested in a parallel study in understanding how patients with systemic autoimmunity respond regarding immune dysregulation to COVID exposure or to the COVID vaccine. But in a really great collaboration with NIAID, with Gigi Notarangelo and Helen Su, and then with groups from a number of hospitals in Italy, in Chile, and in the United States, we were able to look at NET dysregulation, focused both on pediatric and adult cohorts. But what I'm showing you here has a lot to do with pediatric, which was an area less studied in the context of COVID. So we studied two particular conditions, MIS-C, which, as you know, is a very severe form of post-COVID or associated to COVID. And then the other one is what we call COVID-TOS or CLL, which is really seen in patients that were doing well post-COVID, possibly related to COVID, some sort of perennial-like disease, and which appears to be associated with type 1 interferon responses. And so what was interesting is that we found that levels of NETs were significant in the circulation, were significantly increased in the COVID-TOS patients, but not so much in the MIS-C patients from Italy. Interestingly, there seemed to be fluctuations during the disease course, but at some point the levels go down. And we also found evidence of dysregulated NET formation in the tissues from these COVID-TOS kids, suggesting that this may have some role in the manifestations of the condition that we found in these patients. And interestingly, when we looked at them, there seems to be some geographic variation, because when we looked, and I'm not showing it here, but when we look at the MIS-C patients from Chile who were particularly sick, we found evidence that their NET dysregulation was indeed present. And it was associated with the need for more pressor drugs, more cardiac drugs, with more complications such as pneumonia or shock. So somewhat implicated that MIS-C is not all the same, and that there may be some geographic factors or microbiome related, et cetera, that may be associated with how the host responds to the virus, but certainly implicating that or associating manifestations of COVID in kids with NET dysregulation. And in fact, what we found is that in these kids, both in MIS-C and in COVID-TOS, one characteristic feature is that there is impaired clearance of the NETs. And there is a significant proportion of these individuals that did a poor job integrating the NETs. And when we did genetic analysis, it did not appear that they had any predisposing obvious factors such as polymorphisms in nucleases or anything that would explain that. It would appear to be more of an acquired factor during the acute process that then reversed once they got better. That's what we think so far, that these are related perhaps to complement activation and other factors that are transient, but may lead to this impaired NET clearance in the kids that develop these complications. And one interesting last point, and this was done in the adult cohorts, was that we were able to get samples from the initial wave in Italy when the alpha variant. And then what happened with unvaccinated patients that developed Omicron, that got Omicron variant. And interestingly, there was a big difference in the levels of net induction between those that had the alpha versus those that had the Omicron, where nets were significantly lower in the Omicron, irrespective of disease severity. Of course, Omicron is associated with less disease severity, but irrespective of that, it would appear that the Omicron variant has less capability of inducing net formation indirectly or directly than the alpha variant. And this was associated also with the induction of different cytokines, pro-inflammatory cytokines, in close association with the induction of net formation. So more studies are needed, but it may suggest that, you know, not all COVID exposures are the same, and that as the virus evolves, we may see different effects on neutrophil dysregulation, and how that affects disease phenotype remains to be seen. So I will end here just by just summarizing some of the key points I try to make. One is that we think that sex differences in neutrophil biology not still very well understood, but may play an important role in the differential risk for autoimmunity, and the role of sex hormones here remains to be better characterized, but appears to be key. That I would like to emphasize that in the context of autoimmunity, we do believe that nets are an important source of modified, particularly oxidized autoantigens that may promote autoimmunity in individuals at risk, and therefore that targeting neutrophil subsets or aberrant net formation could be particularly important in the context of vascular damage. That is such an important contributor to morbidity in lupus. And then finally, these viral host interactions and how that promotes long COVID and post-infection autoimmune responses in the context of neutrophil dysregulation. I mean, it's an area of great interest that I think requires a lot more studies and multi-center collaborations, and remains to be seen. So I will end here by acknowledging many people that have been involved in these studies, former and current members in my lab, some I acknowledge in my slides, the great support from NIAMS, particularly from John O'Shea, and also all my collaborators at NIAMS and other NIH institutes. And then my great collaborations with both with academic institutions and with some industry partners, and all the individuals, I couldn't list them here, that participated in the COVID consortium we did for the NET studies. Thank you very much for your attention, and I'll be happy to take any questions.
Thank you, Mariana. What a fantastic talk. I think you covered everything you ever wanted to know about neutrophils. I have to say, I got a bit scared about your mentioning of the circadian rhythm. It was already hard enough to study neutrophils up to now. It doesn't make it really easier. So for the audience, again, if you have any questions, just put them in the chat. And we have actually one question from Emmanuel Kawasakis, really nice, his next fellow of us. Welcome, Emmanuel. And he asked whether you checked in your anifrolumab data patients that had a high and a low interferon-1 score, because in the original anifrolumab trial, they responded actually better.
Yes, so the samples we got were a mixed bag, and we did not see, my recollection is that, it's a great question, we did not see any difference in the response to how the nets were modulated between but we may not have had enough of one of the groups to really make a comparative analysis. It is a great point though, but I think what Astra provided was not super selective, because we wanted to have more rather than less samples, but we weren't able to really randomize them based on that. We will have to see, yeah. Yeah,
of course, I think probably to have the large cohorts in this respect is very difficult to obtain. Yes. And then Gabi Kanya from our lab, she asked whether you checked activation of neutrophil phagocytosis in diseases, and if yes, did you see any difference related to sex? Yes, so we haven't looked specific. It's one of the things we want to do, how phagocytosis may be different in females and males. I'm trying to remember if we did any studies or not, but I don't recall we did. There have been, numerous studies in general about lupus and impaired neutrophil phagocytosis from Martin and others. So that appears to be
something that in addition to net formation is happening and may be affecting net clearance. I mean, in the LDGs, we also saw some differences in the ability to phagocytose, but we look very specifically, I think, at Staph aureus or something like that. So I cannot say this is a global impairment. It was a very focused analysis we did. So, but in the context of sex differences, we haven't systematically checked that, and it's a good question.
Yeah, so just also considering sex differences, I found very interesting data. And then Oliver actually posted the link to the Lancet Summit about sex and gender differences in rheumatology. So maybe the audience or you might also find it interesting to listen something there. Then Oliver, I know you also have a question, but the audience is active, so I will keep you on hold for a moment and go again with the next question from the audience, which is, did you go deeper into heterogeneity characterization? So surface markers, for instance, because of course it would be nice to select those by cytometry.
Yeah, so as I mentioned, we did some initial site-seq, and there we were surprised that they all look the same, right? The differences were so small, but it wasn't a very broad panel. So, and it's expensive. So now we're trying to expand the panel to see, do we have some molecules in mind that we think, at least for the type 1 interferon subset, may help us to, we really would like to purify them and see what they do. So I think that's the most promising one, but we need to expand the panel because we are not ready to conclude that there's no difference in any cell surface markers, and therefore it's going to be impossible to dissect further the heterogeneity. So hopefully I'll have more to show next time. Yeah,
it is a challenge actually in all the single cell data to then pick this out and to further characterize them. You have to sort them somehow.
Yes.
So now, Oliver, I leave it to you to ask your question.
I also have a comment, Mariana, because I listened that the influence of hormones is so difficult to study. And at that Lancet Summit, there will be a talk from UCL, University College London, where I think they had a very elegant approach that because they were doing single cell RNA sequencing, I believe, on PBMCs from patients that were undergoing transgender formation.
Yes.
So before, during, and after hormone induction. And of course, that eliminates all that inter-individual heterogeneity that we are seeing because it's only intra-individual at different time points where they are exposed to a very significant hormone exposure. And I found this really elegant. So if maybe one can contact that because, and when they publish, it's anyway publicly available, the RNA sequencing data, because if you have single cell, you could look at neutrophils and really see what the changes are. And listening to your very elegant studies, I thought that might really be interesting for you. They collected, I think, 5-6 patients.
Yeah, no, thank you. And it's funny, every time I give this talk, someone asks me about whether I have studied transgender. And I'm like, no, I would love to do that, but this may give us the answer. We even tried to differentiate progenitors in vitro, exposed to hormone. It was a nightmare. We got such a low yield that I think this may be the way to go. You're right. And if we can get those samples and especially do the modified pipeline for the single cell to really get a better yield of the neutrophils, that may be the way to go. Yeah. Thank you.
And maybe if you have time, no, there's another question. Maybe Günther, you should answer to Günther's question in the remaining time. And my question is not so important.
Otherwise, I will just come back to you, Oliver. So Günther asked, the modified epitopes are the major targets in RA, but usually not in other autoimmune diseases. Did you have a chance to investigate neutrals in early RA or even in at-risk individuals?
Yeah, that's a great question. So the one thing we were able to do, and it doesn't fully answer your question, but we partner with people at the University of Colorado, Mike Hollers and others who have these very nice pre-RA or at-risk individuals based on a variety of, DR4 and other things that they establish, smokers, etc. And interestingly, they do appear to have a higher evidence of net formation and generation of citrulinated autoantigens in the sputum, and in association with both IgA and IgG responses, both in sputum and in the periphery, suggesting that at least dysregulated net formation appears to be happening in some capacity in mucosal sites in individuals at risk. So that's all I can really say right now, that it does suggest that this is an early event. Now, whether it's causal or not, of course, remains to be determined, but that's what we were able to see. Yeah.
Very nice. And then we have a question from Irina, which is a PhD student in the lab. And she said, thank you for the great talk. Related to your study where you assessed the effect of nets on articular cartilage damage, did you also check whether different neutrophil subsets lead to more or less articular cartilage damage, or is this solely dependent on the amount of nets neutrophil produce?
Great question. We didn't assess that. These were very technically challenging studies, and long story short, no, it would be great to do that once we better understand, or we are doing a better job at characterizing these neutrophils. Something I want to say, going back to it's that also I think the level of the different post-translational modifications may be key in how both cartilage and bone get affected. And that's what we're actively studying right now. And as I mentioned, the carbamylation link with bone loss and whether nets express these carbamylated proteins is something we're very interested in studying. And hopefully we're in the process of submitting something about it. So, but we didn't look at subsets either there. So something we need to do.
Thanks. And you inspired a lot of people, as you can see. So Oliver, I think you can give it another try. We still have time.
I'm surprised. There are so many stimulating questions. No, but you know, I think you said that the SLE patients with the IRF5 polymorphism have an increased net formation capacity or something in that direction. And as you know, IRF5 polymorphisms are present in quite some autoimmune diseases, including scleroderma, by the way. So my question is that disease lupus specific, or is that an IRF5 polymorphism effect independent of disease? Did you look at other IRF5 dependent diseases if you see in polymorphism positive patients similar observations there?
So this was actually work from Betsy Barnes at the Feinstein, and it was a paper, I think, in JCI or a few years ago. So she actually looked in people without lupus. So she looked in lupus, but then she also was able to get healthy people who had the polymorphism, who hadn't developed lupus, and yet they made more nets. So it was a nice, of course, it's not causal again, but it does suggest that this is independent of whether they had already developed SLE and have all this inflammation. People that have the polymorphism tend to make more nets. And I think similar things have been seen for PTPN22, for example, and some of the others that, of course, it does many other things, but one of the effects is this increased autoantigen generation by neutrophils. So, but your question is very interesting. Is this relevant for diseases beyond lupus, and can this be something relevant for scleroderma or vasculitis, et cetera, right? And for IRF5, I think the scleroderma question is very pertinent, yeah.
Mm-hmm. Thanks. So we have another question. Mariana, you will be very busy with answering questions again from Emmanuel. And he says, so filgotinib reduces the count of neutrophils very early, so week 1 or 4 in ankylosing spondylitis, the Tortuga trial. Do you expect an association with net formation? Is it possible to measure net in frozen serum or plasma?
Yeah, that's a great question. So, you know, the question I think is also the issue is whether by depleting neutrophils, you still are left with some, right? What's the function of those remaining neutrophils and what are you really targeting? And I don't have an answer to that. But yeah, it is possible to measure nets or net remnants, we call them in frozen serum or plasma. We tend to think plasma may be better when you try to compare it with controls, but again, we've done it in both. You tend to have lower background in our hands when you use plasma on the healthy controls. But yeah, we've done it, and actually it's pretty, as long as you don't do too many freezing thawing cycles, you tend to get pretty good yield and reliable. We have done quite a bit of optimization. So if you're ever interested in the protocol we use, we are very happy to share.
Perfect. Thank you. So maybe I can just, because we have 1 minute left, and it goes a bit along what Oliver said, and we just discussed with the different diseases, because actually what's still a bit of conundrum is that you have similar risk factors in all these different autoimmune diseases, and then also you find this net formation, and I assume that also the nets probably will contain all these histones and nuclear material and everything. So what makes the difference? You know, is it the tissue where this happens? Is it, why then you have one in the joint and one you have in the skin or where?
Yes, I think that's the big question for a lot of things we study in autoimmunity, right? Why if you have IRF5 polymorphisms, or why do you get scleroderma or lupus, etc.
Yeah, exactly.
Yeah. So I think it's probably a combination of factors, right? Genetic, epigenetic, environmental, that, you know, lead someone to have bad luck and develop the disease. And in that regard, you know, the combination of certain factors may lead to neutrophil dysregulation in one individual, but not in another. I mean, I'm very puzzled by this finding that different autoantibodies have different effects on the neutrophil, because you would think, okay, that's gamma related. Well, what is happening here? Why? I mean, we've been, you know, really speculating, why are we not seeing this as a shared phenomenon? So yeah, I don't have an answer to that. I mean, it's a very valid question. It's very important. I think it may be just very context dependent, right?
Yeah.
And as you're mentioning, tissue too, right? What happened, which again, when you look at the circadian rhythm and how the neutrophils go into tissues and whether they're pro-inflammatory or anti-inflammatory, depending on the time of the day where they go, why people get heart attacks early in the morning associated to neutrophil activation, perhaps related to circadian rhythm. I mean, it's fascinating, but I think we just know so little that we will need more time to really figure it out, hopefully one day. Yeah.
So in a couple of years, we just do this again, and then we see how far we came with these answers. So with this, thank you very much to both of the speakers and to all the attendants and to our sponsors, of course. And then I just want to mention that in February, we plan to do another of these online seminars. And this time we look at 3D tissue, in vitro tissue engineering. So I think this is also a very interesting topic that is a lot going on at the moment. So if you like, then join us again as a listener, or maybe even as a speaker. And with that, I wish you a very nice evening, and thanks for joining again. Bye.